I gave you the exactly same problem last time, right? Inside the cell, pump out potassium, pump in sodium, pump in potassium, pump out the sodium, and get the balance. To identify this is me, this is the life. Looks similar, different problem. Read it carefully. 10 more seconds. Last time the question was soap bubble in the air. Outside hydrophobic, inside hydrophobic. You need to have a double layer facing hydrophobic side to the outside, hydrophilic inside. That is the situation of soap bubble we see in the air. If it is in the water, if it forms a bubble, outside hydrophilic, inside hydrophobic, you just need just a single layer of fatty acid facing hydrophilic side to the water side, hydrophobic side to the air side. Okay? Don't just think randomly double by layer at a time. Now you studied about the numbering system, so I changed that word a little. That's okay. Single layer, double layer. Okay, modern life forms, maybe ancient, very old days, maybe it's just a fatty acid, simpler one. But current, the modern life forms of the cell, they use stronger header group, 2 negative charges, and 2 layers of 2 legs, and bilayer of it. So you guys have studied about the numbering, mono, bi, next, mono, bi, tri, tetra, kind of things. If it's a different position, mono, bis, tris, tetra, quis, kind of different numbering system. Okay, about the 4 nanometer, the chain length of a carbon number is approximately 20, one side, and double layer 0 to 20. Carbon-carbon bond length is about 1.5 angstrom, but that's a zigzag, so that's why length may be about the 1 angstrom, that's a 0.1 nanometer. So 20, 20, 40, about the 4 nanometer. It's good to know, and you can compare of different particles, different proteins, how they, how to compare those sizes near to the cell membrane. Our cell is about the 10 micrometer, bigger one maybe 20, super big one, the biggest one, egg cell, 150 micrometer. That's exceptionally big one. The regular size of cells of approximately 10 micrometer size. That's also very good to know. Bacteria 1/10 of our cell size, then volume-wise it should be 1,000 times, right? length is the 10%, then volume is three-dimensional, about 1,000 times smaller. Volume similar, weight maybe also similar. Mitochondria is like bacteria. So bacteria is 1 micrometer, mitochondria longer, less, but anyway, by average you can think about 1 micrometer size. Then chloroplast, lemnochere also about 1 micrometer size, you can assume that's originated from bacteria. Virus is another 10% of a bacterial size. It's easy to memorize. Animal cell, plant cells, mammalian cells, about the 10 micrometer size. Bacteria, 1/10 of it. Virus, another 1/10 of it. Okay, approximately. Of course, there's a bigger, smaller one, but approximately. That's good to know. Now I use intentionally different terms. Small, high, single, double. Okay, good. Mitochondria, chloroplast, nucleus membrane, they have 2 layers of a bilayer. Okay. We will see those structures soon today. mRNA is synthesized inside the nucleus and then exported to outside the nucleus. Protein is synthesized by ribosome, but ribosome is localized on ER membrane usually. Okay, usually the post-translation modification, after translation modification happens in ER, in Golgi, and they are modified. Not bad. Okay. 유리가 제일 빨랐잖아. 축하해. Okay, today we are going to study about the structure of cells. So last time we thought about the hypothetical model of an ancient life. First of all, the life forms should define its boundary. Outside is the environment, inside may be the organized structure, so-called life. Self-maintaining, trying to reduce the entropy and replicate. So those kind of unit we define the life. And modern form of the life. Well, when I was a middle school student, usually we use this as the model of the cell structure. It's easy to make it layer by layer. And then if you take those cells and then stain, they look like this. Actually, this picture was drawn 19th century. It's amazing, isn't it? That means they had a microscope, they knew about the nucleus, and then they knew about this structure. So this structure, what do you call? Those condensed DNA structure, what do you call? Chromosome, 염색체. Chromosome, chromo means color. 종 means it's body. Colored body. It's the same meaning in Korean. 영색, 책. That's exactly literal translation of chromosome. Stained body. That's a chromosome. So that means even 2 centuries ago, not exactly 2 centuries, but we are living in 21st century, 19th century, they already knew that cells are dividing, chromosomes are condensed, they lie in the middle of the cell and divide into 2 kind of things. By the way, by looking at this, is that possible to see from these onion cells, so many cells are dividing together? Maybe not. Maybe 1 out of 100 or 1 out of 1,000, those cells may be dividing. Maybe this is exaggerated. Observed one of this. He didn't want, maybe the scientist didn't want to draw 1 cell out of 1,000, so he collected those observations into one picture maybe. Anyway, with this picture, you can catch the idea, chromosome is easy to be stained. If you add any colorful material, they can stain those nucleus. And based on this, we did some model of the cells. And we'll figure out later how this kind of structure work, the design. If we redraw into 2-dimensional the model. By the way, can you guess what software did I use to draw this? PowerPoint? No. Photoshop? No. I used the CamDraw. So even using simple CamDraw, you can draw. It looks like okay, the picture like this. Anyway, based on this structure, first of all, is membrane. Membrane structure outside the membrane, this is a single bilayer. One bilayer means it's facing 2 sides. And phospholipid, head group has a phosphate, has a strong charge. Carboxylate has only 1 negative charge. Phosphate has 2 negative charge. So that's why that separating effect, hydrophilic effect is stronger. And 2 tails rather than single tail. To carry these tails, they use this kind of linker, so-called glycerol. Glycerol has 3 hydroxyl group, 1, 2, 3. 2 of them is connected to fatty acid as ester bond. And then one side is connected to phosphate group. So this is a much stronger detergent compared to fatty acid. Fatty acid has 1 leg and 1 tail and 1 header group. That's 1 negative charge. Now we have 2 tail and then 1 stronger header group. So this separating effect is much better. Okay? Using this, we define our cell boundary like this. If you define this way, then you can say this is outside, this may be inside. Now we are going to look at the inner the structure inside there. Now this is one problem I gave you to give you an idea about the speed of light and how fast it can penetrate the cells and also the palm city. One of you gave this kind of calculation. Last time I said 10 kilometer, but this student put the 30 kilometer. That's fine. If that condition is not properly given, then you can define that. Even though that's a 10, 30, error range is only the 3 times. That's okay. Now by this, many of you reached up to here and up to here. You didn't finish up. Hey, in numbering system, when I gave you that online the lecture, I told you we don't want to say 10 to the minus 14 or 0.0001 kind of things. We wanted to convert that into tangible number, 100 or 30. Okay? From now on, try to use this tangible number between 1 and 100 range. That's much easier to memorize rather than 10 to the minus 14 or 0.001 something. Okay? So that's easier to keep in mind the keeping the range. So this is approximately 1 cell. It's 30 picosecond. It's quite fast. It can penetrate the light. And then now let's think about the membrane. Membrane, now first of all, they designate the boundary of my space and our space. And like the cell membrane, usually they carry cell membrane potential. Most of the cells outside is a positively charged, more positive than inside. Now, if you look at those composition of this membrane, what is the component of the membrane? Phospholipid. If you look at the structure of the phospholipid, then it has a tails, hydrophobic tail, and it has head group, charged head groups. If you look at these functional groups here, do you see negatively charged one? Head group? Do you see neutral? Do you see positive charged one? How about some of them? This is negatively charged, -1. This one minus, plus, minus, altogether minus. This one -1. This one minus, plus, neutral. Minus, plus, neutral. Minus, minus, -2. So some of them is neutral, cancelled out. Some of them is -1. Some of them is -2. There is no plus here. So that means in both sides, inside and outside, they have a little different composition. But eventually, all of them should be minus. Then how could you get the plus here? This means most likely inside is a little more negative than outside. So relative potential wise, outside is less negative than inside. That's why as the net effect seems like relative potential across the cell membrane, outside is a little more positive than inside. Okay. So to maintain this, now always we need to pump out potassium. This is inside, this is outside. Pump out potassium all the time, and pump out sodium all the time, and pump in potassium all the time, and then the relative concentration of sodium to potassium is almost similar, 100 millimolar concentrations, to keep osmotic pressure balanced. If not, cells will be swollen and the burst, or it can be shrinked if the charge balance is different. Okay? So through this membrane, eventually, basically, the charged molecules cannot penetrate easily. That's why to pump out or to pump in the potassium, pump out the sodium, we need to utilize either channel or pump. If it goes across against the gradient, pump in the potassium, you need to use the energy because this is higher inside. Then you need to use a pump to pump in. If you open channel, then potassium will be moving this way because it is a higher concentration here. Okay? Channel is a passive way, faster control, and pump is using energy to move one by one. Okay? By that way, we segregate those two spaces, and then we balance those materials. Now, to do that, on the membrane, there are a lot of proteins that are localized, and those proteins, some of them is enzymes, some of them are pump, or some of them are channels. Those are the functionally the control those metabolites in and out of those material and changing those materials using different chemical reactions. Now, among this the membrane structure, this is a so-called mosaic structure of the membrane. Sometimes these proteins are not randomly scattered. Sometimes they are collected together in some area, quite a rigid area. And to make that membrane rigid, you can see this kind of structure here. These are cholesterols. Lipid molecules are defined by its physical property. Right? Lipid molecule is defined as hydrophobic molecule. Naturally occurring hydrophobic molecule is lipid, and they have a tail structure and they have a steroid structure like a cholesterol. Now this membrane, if you add cholesterol, then this is lipocholesterol. Membrane structure can be flexible. We have this kind of wall Now, if you add this kind of stone in between, then make this wall more rigid. This is roll of cholesterol coming here. And this relatively rigid area, and they can anchor different proteins in this area. So cell membrane is not homogeneously scattered structure. They have this membrane, the membrane proteins getting together to make some domains. That domain is called as lipid raft. Raft is a demo composed of lipid. So they get together, and then this area they have a high cholesterol. Means those area is more rigid structure. You have seen those the video clip, and looks like some domain was floating around in the water. So that water is like this membrane, and this whole domain is moving around. And those domains contains a lot of receptors, channels, pumps, kind of functional proteins of them. Okay. So this is kind of model, more realistic model than these mosaic structures. Now, cytosol, inside cell. Usually we have the concept of this is like a proteins are swimming around the cytosol, kind of image we have. So these are proteins, functional materials, and then cytosol is like water, but actually our cytosol is highly condensed. It's more like these proteins are contact to each other. It's more like the marine is the swapping in the mud kind of situation, not swimming, because they are so high concentrations. There is almost like, if you look at those, the material concentration, 400 milligram in 1 milliliter. That's almost a half solid in the liquid, isn't it? That's really half water, half fish kind of situations. So it's a highly dense of the materials. Inside the cell is not so swimming area, it's a very crowded area. But for convenience, we have the picture of proteins are moving freely, but actually to move the protein moves, it needs to squeeze out the crowded protein environment. So this is the cell environment, cytosol. Now let's think about another structures inside the cell. The outside membrane is bilayer, one single bilayer. So that defines outside and inside. If there is vesicle, small, another one inside, one way it can form is endocytosis. So this outside membrane structure can be moved inside like this, move in, and then isolate this membrane, and it forms another membrane-bound structure inside the cell. Okay. So this can be derived from ER or Golgi, or from plasma membrane inside. Then one membrane structure outside, another membrane-bound structure inside here. Now, if you think about the composition inside this material, inside this vesicle we call endosome. Endosome means it was formed, it's a body through endo. So this body was formed by endocytosis. From cell membrane, it is moved inside, and then it forms this kind of vesicle. We call them endosome, and those endosome can be matured to form lysosome, kind of a vesicle structure. This vesicle structure is similar to a plasma membrane. It's a lipid bilayer, the single layer. But composition of inside here, this inside is comparing to this is environment and this is cytosol. Right? Inside here, this environment is similar to cytosol or similar to environment. If you look at this form here, inside of this endosome is actually from outside. Right? That's why endosome's inside is actually its outside material. It is formed inside the cell, but we utilize this endosome usually for garbage treatment to destroy by making this environment acidic, and then destroy all other garbage material inside here. It treats like trash box inside the cell, and inside this environment is more similar to outside, not inside. And this process, if you reverse, then that process is called exocytosis. Makes sense, right? So coming in, endocytosis. Moving out, exocytosis. So that's the direction. Now, endosome is formed like this way. Then how about how to form exosome? Endosome is forming those vesicles inside the cell. Exosome is moving out to outside to make the bubble, like a vesicle structure. That exosome can be formed by exocytosis. You think? Think about it. To form the exosome, exocytosis can make exosome. Not clear? If I draw this way, this is cell membrane. Now this was endosome. Endosome is moving to the cell surface. If we touch the cell surface, membrane fuse, and inside the material is secreted to the outside, right? Where is the exosome? Exosome is not formed. Exocytosis is a reversible process. Reversible process of endocytosis, and it doesn't form this kind of outside the vesicles. It is just a fuse in. If this moves to the surface, go in and fuse, and then only inside the material. This inside is originally similar to the outside material. There is secreted to the side, the outside of the cell, but there is no extra vesicle formed to the outside. So exocytosis is not the answer. The process to form exosome. Right? So here we need another the process. Now this here this is inside the cell, and this is forming the bulbing out to the outside of the environment, and then eventually this form like this. This is kind of reversed form of endocytosis, but that reversible process we call the exocytosis, and this is a different process from that. So this process we call a little different term, ectocytosis. Through ectocytosis, now we can form this kind of exosome. Exosome is kind of a hot word these days. Extracellular vesicle or exosome, this kind of the material can contain genetic information and a lot of components in there. It can be transferred to the long distance, or it can touch other cells. So this exosome is quite important. Now, question: this exosomes inside, is it like inside or outside? Here the inside means inside of life, outside of life. Exosomes internal material is like a cytosol or like environment. It's like a cytosol, isn't it? So this was white material here, cytosolic, budding out, and then it forms this vesicle. This inside the material is similar to the cells. So that should be considered as an inside. Right? So it's like a small cell is separating and moving to the different environment. And then if you fuse with a different cell, it's kind of fusing two cells together to form one cell. Okay. So two different the process we said. So one is endocytosis, and then the other one is exocytosis. Exocytosis is usually used for secretion of the material through membrane fusion, and then ectocytosis is to form new vesicles, so-called exosome. Okay, now let's think about the protein synthesis. Nucleus here, mRNA is synthesized inside the cell by making small copies of DNA into mRNA, and then that mRNA is exported to the cytosol through nuclear pore. And then here is the endoplasmic reticulum structure. Reticulum means it's kind of a network, and here this space And inside the area, these dots are, they are ribosome. Ribosome is the machinery to make a protein. So they have this kind of a dot, looks like a rough. So this is a rough ER, they call. And outside they don't have the ribosome, it's a smooth ER. So then if I ask you which one is more inside, yeah, rough ER should be closer to the nucleus. It is a protein synthesis part. Once it is synthesized, and then those proteins are modified first in smooth ER, and then outside here they have another body, so-called Golgi body. They have another modification. The process at the ongoing. Okay. Now let's think about the protein synthesis. Ribosome synthesis. Ribosome. What is the component of ribosome? Where is the ribosome is synthesized? Ribosome is synthesized inside the nucleus. Ribosomes, the component is? Ribosome's main component is RNA, not protein. Ribosome is a kind of enzyme, maybe which is the most important enzyme in the body, making protein. Our body is the dual system, right? We have an information system in DNA, and those information is transcribed and translated into protein. So we have an information DNA system and protein machinery system. We have a dual system. So making protein is one of the fundamental, the function of life, our life. And interestingly, most of those enzymes are made by protein. But this very important fundamental enzyme is made by RNA. Maybe in ancient times, we didn't have a separate DNA protein. Maybe we had an RNA as a genetic information, and RNA as a functional material too. RNA is very, compared to DNA, super unstable. That's why they can change very quickly. We are observing those fast changing, fast evolution through RNA virus, like the coronavirus. They are unstable, but they can change very quickly, and they have a chance to make more diversity very quickly. Goes through the evolution pathways much faster than DNA life. And using this can make ribosome inside the cell. And then protein synthesis is happening outside the nucleus. Right? That means mRNA should be transported to the cytosol. Ribosome also should be transported to the cytosol. And ribosome anchored on ER. Those ER we call rough ER. And they take mRNA and start to make the proteins. And those proteins, they needed to go to the everywhere inside the cell. Cytosol, membrane, and different organelles. Then how do they sort out to send which protein to where? That's a good question. And after that, protein is composed of 20 amino acids basically. But those does not provide enough diversity. Time to time, they need to modify further by adding acyclic group, lipid molecules, carbohydrate, those kinds of modification we call after translation modification. Means non-protein motif. They add a different motif to the protein. That's post-translation modification. It happens in ER, especially smooth ER, and also in Golgi, and then modify those structure. Now let's think about those localization. How they can decide those localizations. Maybe one way. So this is nucleus, and this is ER, and ribosome is here, and this is the mRNA. Ribosome localized somewhere on the surface of the ER, and the protein synthesis happens. So this is reading mRNA through here. And then protein synthesized is moving out this way. If this is localized in this topology, then what is the end position of protein? It should be cytosol. So this is one simple way. If it's a synthesis of a protein happens to the cytosol direction, then this product will reside in the cytosol. After that, maybe depending on those signaling peptide at the end, then it can decide to go to lysosome or mitochondria or different other organelles inside the cell. That's one way. So ribosome is just to synthesize those protein to the cytosol directly. That's one topological approach. The other way, it is possible that you have seen some movie in the video clip, and the synthesis can happen and secrete those protein inside this lumen. I draw this lumen this way. This is ER, right? And then this is an isolated ER structure. And if you look at this inside here, lumen of ER is inside or outside of life? Inside? It's outside. Inside is the cytosol, is inside, right? That's our reference point. Cytosol is inside. Then this endosomes inside is outside. And lumen of this ER, same thing for Golgi lumen is outside environment, right? So that's why this is synthesis. Previous case, this is the synthesizing those protein secret into the inside space, cytosol space. But sometimes, depending on those orientation of ribosome, and maybe depending on this mRNA sequence, they synthesize this protein and secret into this space, maybe into this space, and then it forms this kind of an endosome. Then, and the result is this vesicle is moving to the plasma membrane. One of you described it quite well in this question. And synthesis happens through this description, is basically describing this process. Protein is synthesized and secreted into lumen of ER, and it forms its vesicle. Protein is now traveling one by one to the plasma membrane here. Usually they are packaged in the vesicle, and this whole vesicle was moving to the plasma membrane. Right? Who was the carrier of this, the vesicles? There was the machinery of working, the carrying those vesicles and working to the plasma membrane, right? That's a real motor protein, so they are moving like this. And that was quite a hot topic like 10 years ago to image those movements. And then what was the train rail? That was a microtubule. Microtubule making those network from inside to the outside the cells. And we have two basic cytoskeletal structure. Microtubule is just sustaining those structures. It's from center to the cell membrane, sustain like this. If you sustain only this way, then cell will be burst. So outside there is another cytoskeletal structure to push it out to the inside. Those are mainly actins. So actin skeleton and microtubule structure is balanced out to maintain the cell structure and shape. Okay. So now, if the protein is secreted into this surface, this space, then eventually this vesicle is moving to the cell membrane, and eventually they fuse into the cell membrane, and the position of these proteins are outside of the cell. These are secreted proteins. Maybe signaling the protein or hormones, kind of proteins are synthesized into this lumen. Okay, so that was the question I gave you in the quiz, and also that is the description here, how they can be the traveler. So the case. Actually, this is the movie. If you look carefully, the video clip, it was synthesized into the lumen, but at the end position, there was anchored on the membrane. If there was anchored on the membrane, then what happens? The end position is this material is moving inside here, but anchored inside here. This is the inside space and this is the outside space, right? If it moves to plasma membrane, then what is final topology of this protein? It's facing outside, but this is anchored on the membrane. Through this process, what process is this? This is the endocytosis, exocytosis, ectocytosis. What is it? This should be exocytosis, right? It's similar to fuse and then secretion, but this protein was bound here, and the main protein part is facing to the outside. So this is kind of anchoring, and then showing those main part of the protein is to the outside. So this is very simplified version, but our cells are making thousands of, maybe more than 10,000 of different proteins. And by modification, you can generate maybe millions of different compositions. And then those, how they localize into final positions, these three are kind of representative schemes, whether to the cytosol, or for the secretion part, or membrane-bound proteins. Okay. Now, cellular structures. Now let's think about seriously about the in and out. Okay. Now, this is outside. This is inside, we said. And here, Golgi, and ER, we already defined. Lumen means it is the inside. Lumen of ER, lumen of Golgi, they are outside. Okay? And I gave you some question like that. Seems like the question was not very clear, so sorry about that. So what I meant was, the question is whether which part is more like life space versus environmental space. That is the question. Okay. So life space we call inside. Environmental space is outside. If we define that way by looking here. So outside here, outside here, outside here. Inside is the cytosol, is the inside. Okay. Now let's look at the mitochondria. We said we already answered or memorized and understood hopefully. And mitochondria has a double layer of membranes. Outer membrane and inner membrane. Outer membrane, phospholipid bilayer. Inner membrane, phospholipid bilayer. It has two layers. Now let's assume that mitochondria was independent bacteria a long time ago. They came into our body and reside inside our cells. One way is maybe coming in and fuse and just become one cell. But seems like they didn't choose that way. They have their own space here. They have their own DNAs. And they have double layers. Outer membrane, inner membrane. This space here, they call matrix. And then this space, intermembrane space. The name, the designate what that is. Now, which part is inside and which part is outside, do you think? Let's think this way first. Matrix. Is the matrix inside or outside? Then, intermembrane space, is that inside or outside?
Outside.
Maybe that's not the case. So that means mitochondria live in our cell, but they have this kind of intermediate space, and this is similar to outside space, environmental space, and they have some boundary, some area in between. So this is the inside of bacteria. This is the inside of us, and this is kind of DMG. DMG 우리말로 뭐니? DMG를 생각나고 우리말로 비무장지대. So this is a kind of a buffering space in between. We are keeping outside the space in between bacteria and our cells. That is the, seems like that is the structure we see. That's how we call this as double layer of phospholipid. Now, let's think about the photosynthesis one more time. Photosynthesis, utilizing sunlight, and we take water as the material. Also, another material source, carbon source as gas material, CO2. Most stable, most oxidized carbon material and then utilizing this energy, and then it generates this kind of energetic material. And this energy material eventually it make sugar molecules, and this is light reaction and this is dark reaction, two-step reactions. As an then byproduct we generate the oxygen, and this is a summary of photoreaction, the photosynthesis, carbon monoxide, carbon dioxide, and water, garbage material, and just environmental material, and utilizing light as energy source. And then it constructs carbohydrate, very useful, the energy storage material, and also our life material. And byproduct, oxygen was generated. So for ancient the time, maybe we didn't have a high oxygen concentration in the air. Because of this photosynthetic pathways, oxygen is accumulating. Now in the air we have 20% of oxygen. Oxygen, in a sense, very toxic. 산소 같은 여자, very toxic 여자. Woman like oxygen is a very toxic woman, because oxygen is a catchphrase of the cosmetics, but oxygen is a very toxic material. It's accumulated in the air, and to utilize that, let's think about the photosynthetic pathways first. Photosynthetic pathways, first thing is capturing those light energy. I briefly mentioned the last time, there are light capturing machineries, so-called photosynthetic machineries. They capture this energy and they excite this electron. That becomes a hot electron, high energy electron. That energy is stored first into this kind of material here, and then it is converted into this. And electron energy is step by step, cascade, step by step, giving some energy, and another one, and fully utilize this energy and generate these materials. And the position over here is eventually intermediate. They generate proton gradient. High concentration of a proton here. This is the inside of chloroplast. This is the outside. Chloroplast, similar to mitochondria, it also has inner space and intermediate space. Intermediate space, they pump out proton. Acidic environment is not healthy, so they pump out this proton, but this utilizing this proton, they synthesize the ATP like this. This happens in chloroplast. And now here again, outer membrane, inner membrane, and if you look at carefully, inside here is bacteria's space, outside here, our space, and there is some intermediate space, very similar to mitochondria. And then utilizing this, then final molecule they make is ATP. And ATP, if you look at the structure, diphosphate versus a triphosphate, it's similar to anhydride structure in organic molecule. There is more energetically high energy material. So here, this bond, they store some energies. That's ATP. Before ATP, they make this kind of material, NADPH or NADH. Basically, they are almost the same material, except phosphate group is here, then NADPH, and no phosphate, the NADH, almost the same, right? Now, they store their energy inside here. Where do you think they keep their energy? In ATP, they had energy here, right? Diphosphate versus triphosphate, and energy was stored here. When they use this energy, they keep this phosphate and release that energy and utilize. NAD, NADH, NADPH. If you look at, there is diphosphate here. Maybe that's maybe candidate you may consider. Actually, the real situation is they utilize this mode. This is the oxidized form, and this is the reduced form. Through this reduction means taking electrons. And how many electrons in this case? 2 electrons. That electron is generated by photosynthesis, and then activated the electron. 2 of those electrons are transferred to this molecule and reduce this. This is a condensed energy stored inside this molecule. So NADPH is oxidized form. reduced form is H, NADPH. One NADPH can generate 3 ATP. So that means NADH or NADPH is a higher energy material. First material was excited electron, hot electron. That was transferred to this by reduction, and then make NADH. And then NADH utilizing this pump, NADH, NADPH, they utilize this pump, they generate ATP. So ATP is, energy-wise, is the lowest energy. NADH can make 3 ATP, and then this electron can generate a couple of NADPHs. So hot electron is the highest energy, and NADH and ATP. Now this happens in chloroplast. Very interestingly, if you look at mitochondria, almost the same machineries are found in mitochondria. Usually we believe that mitochondria is energy generation part. Right? Where does that energy come from? It's by breaking carbohydrate. Breaking carbohydrate and generating fuel for TCA cycle. And through this TCA cycle, they generate NADH, which is very similar to NADPH. And then they transfer this NADH energy to generate this proton gradient. Sounds familiar, we just have seen in chloroplast. Right? And this machinery, ATP synthetase, utilizing this proton gradient, pump in, they generate ATP. By the way, this machine and found in chloroplast is almost the same machinery. Chloroplast is maybe part of plant cells. Mitochondria, maybe animal cells, and also plant cells too. Maybe they evolved together and they share a lot of common fact here. Not just the metaphor-wise is similar, it's exactly similar machinery they use. In photosynthesis, they capture light energy and excite electron, and utilizing that electron, they generate NADPH. In mitochondria, already stored the material like glucose. By breaking up that glucose, they generate NADH. And utilizing this NADH, they generate ATP. It's very similar to each other. So that's why photosynthetic pathways to make glucose. And breaking up glucose generating energy, if you draw side by side, there's a lot of similarity in between.
You said that it generates ATP by pumping in hydrogen, but the hydrogen concentration gradient is higher outside in the intermembrane space, right? So the ATP synthase, is it more like a channel protein or?
It's like a channel protein. It's more like a pump protein. It has a dual functions. This one, now here, this is intermediate space and this is inside of the bacteria. If you look at the chloroplast, it's a similar situation. This is our side, and this is the bacteria inside. This is bacteria outside. So same way, proton gradient is happens here, and then pump in and generate ATP. Now this is kind of a pump protein. If we have enough ATP, utilizing ATP is a pump out. If there's a high the proton concentration outside, then utilizing that gradient, it's a reverse reaction, generate ATP. So pumping out is a pump. Coming in and generate ATP, we call ATP synthetase. It's same reaction, reversible reaction with a different direction, we give the name. Now, then, other photosynthetic bacteria, how do they do that? They have only one membrane. They don't have this outer membrane. They have this membrane, right? And really, similar the pattern here, if they do photosynthesis, they pump out that proton to the outside and utilize that proton and generate ATP, they do. In the case, you can imagine that maybe this proton gradient cannot stay there for long, right? Pump out the proton, they can be diluted out to the environment. That's why photosynthetic efficiency is not so good. Once they are inside our body and they have this outer membrane, then they can keep this proton concentration high enough in this space. Then they can utilize that proton gradient more efficiently. In the sense, now if you look at this the mitochondrial space, we just simply memorize that, oh, mitochondria has a double layer of phospholipid. But if you think again, inside is a bacterial membrane, right? Outside should be a membrane, like endosome or ER kind of a membrane. And if you look at those protein, the localized inside on this membrane and on this membrane, you can see the difference. They have a more bacterial membrane protein here, and outside they have more proteins there. So, if you isolate the mitochondria, it looks like it has a double layer, but actually we are capturing those bacteria inside our membranes structure. That's the mitochondrial structure. Interesting. Okay, based on this, now let's just think about nucleus. Nucleus is also double layer, we say. Then, question. Inside of mitochondria, this matrix space is a bacterial space. It's inside, but not our inside. That's our partner's inside. Could be enemies inside. Right? We wanted to utilize mitochondria as a function, but we don't believe it totally. That's why we just isolate it out using our membrane here. How about our nucleus? Our nucleus is the space of keeping our genetic information. That is key information, key material, key space for our identity, isn't it? If they use a deep similar structure like this, then inside of the nucleus should be somebody else's space. That doesn't make sense, right? So now if you look at those nucleus of the space, nuclear membrane, maybe it was made from, let's say this is like ER structure, and ER structure is kind of an extended plasma membrane into the cytoplasmic space. And then they come together, and then they generate this kind of isolated space. And the result wise, here this is outer membrane, this is the inner membrane. But if you look at only this is a part of this. Outside, inside, both of them are our membrane. And in a sense, this is not our cell membrane is topologically it's isolated space, right? Isolated membrane. We can define in and out. But if you look at that means this space and in the inside of the nucleus basically connecting the same space. Right? So that's why if you look at the nuclear space, nuclear membrane is more like surrounded like this double layer, but double layer is both of them are like our ER membrane. Both membranes are our membrane, and they have a holes. This hole is quite big hole. Small molecules are easily can get in and out. You don't need pump or the channel. That's what we call nuclear pore. And through this space, materials can get in and out. And this space, again, inside of the nucleus space is our space. Then it's safe to keep our genetic material inside the nucleus. Now, another small other part inside the nucleus, you've been mentioned previously. This is a so-called nucleolus. In Korean, that's in. Nucleus is the head, right? So this is the center of ribosomal RNA synthesis. So they have a ribosomal DNA here, and using ribosomal DNA, they make a lot of the ribosome. So ribosome synthesis is happening here. Very interestingly, this nucleolus structure is not segregated by membrane, but this is kind of a separate phase, different condensate. Looks like it forms separate phase, but it doesn't have a membrane. This kind of a structure now getting more and more attention in the modern biology. So far we have seen lysosome, ER, mitochondria, they are all separated by membrane structure. But this one has its own space, but there is no boundary. It's like the oil drop inside the water. So it has its own separate phase, maybe different the chemical physical property of those components. The high amount of RNA and protein complex, they form separated unit, and we can visualize it, and that is what we call nucleolus, but that is not membrane, membrane mediated separations. It's a separate phase separations, physical separations. So those kind of condensate. So we have known nucleolus for a long time, but recently we started to see those kind of condensate inside the cell too, here and there. So those can be quite the important new topics to study. What is their role and how they are connected to disease and to delivery kind of things. I think this is the last one. Okay, any question for this structural cells?
So ribosomes are synthesized in the nucleus. Is there any like the mRNA for protein synthesis needs to go through the post-translational modification before it can be transcribed into a protein? But is there any sort of overlap between the two since they exist in a similar space?
Okay, a little confused here. So ribosome synthesis is a protein synthesis machinery. So first of all, it should have a lot of DNA inside here. Based on the DNA template, they make the mRNA. So that's a separate enzyme, so-called RNA polymerase. So they make mRNA, and mRNA is a transport to the cytosol with this 4, and they move to the outside. That's the information system. Now, this position is making the machinery to make the protein translation part. Post-translational modification means after protein is synthesized and then modifying. So that's a later stage. mRNA is synthesized by reading DNA. That's happening here. And then mRNA maturation also happens by removing intron and connecting exon and other things. That's a separate event here. Those two are independent. So one is making mRNA mature, going out, and then wait for synthesis. Ribosome is synthesized here, and then they move out and attach to the ER, and they wait for their mRNA, and then they meet together and start to make the protein. So first step here is the transcription. Here, translation. After protein is made, then they move to ER, Golgi, plasma membrane, and slowly modify. That's post-translation. Other question?
That was the
Exosome.
what is the driving force to form ectosome structure?
Driving force for?
Ectosome structure.
Exosome.
Ectosome, the slide, yeah, that.
Ectocytosis is the process like this, forming this exosome. So this is, the process name is ectocytosis. And then the product is the exosome.
So my question is, what is the driving force of forming the exosome structure?
If the cell need to send out some material in highly dense form to neighboring cell, maybe they start to utilize, collect some motor protein around here. It's not automatically happens. There's a protein cell collected together, and they start to squeeze out to make this bubble to the outside. And then this side is eventually squeezed and then click, and then release. So this is a highly designed and also energy consuming process. So it's not natural driving force like the gradient of the ion kind of things.
So the cleaving of exosome is done inside the exosome structure.
If you think the ectocytosis in a similar way to the endocytosis, in this the way also there is some proteins should be gathered together and then they squeeze out and cleave. And I showed you only one example of ectocytosis. Actually, there are several different kinds of the method. Sometimes there is a small vesicles inside here, and sometimes they're forming this way. If it forms this way, this is very similar to endocytosis process. Then there may be some autoproteins that come together, and then they generate this kind of structure and then release. 마치고 다시 한번 마치고 명확하지 않으면 움직이는 중입니다. Any other question? Great.
So some proteins are secreted directly into the, and some are just secreted into the cytosol. So is there like a difference between those proteins?
Definitely, the some proteins they need to go outside as like a hormone, and they are synthesized into this lumen and then secreted. If there are the machineries of an enzyme which is important inside the cell, then they should be generated in the cytosol. How? That's a different question. And maybe the mRNA sequence or the mRNA binding part has some difference, and then maybe the ribosome may have a differently oriented ribosome. Maybe some ribosome is facing here and then generating here. Then they may have another accessory proteins which can take mRNA for intracellular machinery. Then they can synthesize the machinery to the cytosol. Some other cases, this is a secretory one, then they have different sequences here, maybe. And then they take it and then secrete it inside here. So how to regulate those function location is maybe talking about the further study, but we can imagine there may be different controls. Some ribosome synthesized into here, some ribosome synthesized to the cytosol. Okay, based on this, the next week the study material is molecules of light. So today we just show those topological positions in and out, and protein synthesis briefly, and DNA replication, transcription, and translation. So next week study material is a little more details on how DNA and transcribed into RNA. What's the different structure of DNA RNA kind of study. So that's molecules